Welcome to chapter 3 in the human biology course. In this chapter, the author discusses essentially what kind of cells exist in living organisms, and we're going to go over different varieties of cells, kind of a general overview, and then go into specifics about animal cells, because these are the type of cells that we're interested in the most in this course because we're studying human biology. Now, first way is to generally categorize all the cells. So we have primitive type of cells called prokaryotes versus more advanced, more complex type of cells called the eukaryotes. When you see these words, the root word of the karyo essentially refers to the center, the most important part of the cell, which is the nucleus. That's where the genetic material is stored, such as the DNA. Now, for the primitive cells, they actually do not have a distinct nucleus, although they do have their genetic material essentially kind of free-floating inside the cytoplasm, the inner compartment of the cell. And for the eukaryotes, they have the true nucleus, very distinct nucleus that you can see under a microscope, and all plants, animals, fungus, and in general most cells other than bacteria will be of the variety of the eukaryotes. Now again, as a basic overview of this, for the prokaryotes, essentially you're just thinking about cells that are primarily bacteria, unicellular or one-celled organisms, very simple, very primitive, one of the earliest forms of life on Earth. These cells always have a rigid cell wall to protect them from the external environment because they live not as a multicellular organism, but again as a unicellular, so they need extra protection against the environment, against weather and other changes in the outside world. Again, as I said before, they do not have a distinct nucleus, or they do still have the genetic information inside the cell. In terms of size, usually bacteria is much smaller than animal cells or other cells that we're going to talk about here. So that kind of a scale change is very important as well to consider. Now for the eukaryotes, again, our animal cells, all plant cells, all fungi are eukaryotes. These are the most advanced type of cells, the ones that came later after prokaryotes, and these are the cells, essentially of the variety that's found in our body. So our cells have a cell membrane, but never have a cell wall. Now in plants and fungi, there is a cell wall, although if it's of a different variety than in bacteria. This is mostly just to consider that, keep in mind, plants and fungi are essentially more exposed to the environment and sometimes do survive as essentially unicellular organisms to some extent, or at the very least are exposed to the elements, to water, to rainfall, to sunlight, things like that, much more so than us, and so need that extra level of protection than animal cells require. So again, in animal cells, we do not have a cell wall, although it is found in plants and fungi. Very important characteristic again for eukaryotes is the presence of that distinct nucleus of the DNA, the genetic material inside that codes for all the molecules and all the functions that the organism will have. Eukaryotes always have multiple organelles. These are the little organs, little structures inside the cytoplasm that will allow the cell to undergo its normal functions such as reproduction, metabolism, digestion, and so on. That we're going to talk about very soon. A very important component of every cell, regardless of whether this is bacteria or prokaryotes, eukaryotes, or any others, or our animal type cells, is the cell membrane, or what we call the plasma membrane. Now, plasma membrane is that outer boundary of a cell that protects the cell from the external environment, but it's not rigid like a cell wall, but it's more dynamic, right? It's more fluid, it's changing. It is allowing molecules to enter and exit the cells when appropriately under the methods of selective permeability, which we're going to talk about soon. Now, when you think of the plasma membrane, you're essentially thinking of a phospholipid bilayer. This double layer, right, that's shown kind of here. Imagine that you have a cell in front of you and you magnified one small portion of a cell membrane and brought it kind of to this image like this here. So you have your double layer. This is one part of the layer, and this is the second part, right? So bilayer means double layer. Phospholipid refers to the molecular structure of the main components of those layers. So when you see these red circles, this is the phosphorus component, and the little blue squiggly lines, that's the lipid component. Lipid, remember, essentially means fat, okay? And phosphorus is the component that is resembling water, right? So one is part that loves water, and the other part of the molecule hates water, right? Because remember, fats and oils do not mix, right? Like oil and water doesn't mix together, so they repel each other. This is a very kind of interesting combination of chemical interactions that we get here. So because of this, essentially everything, let's say, imagine this is the outside of the cell, this is the inside of the cell, right? So everything that's facing outside of the cell and inside is water-like. We call this hydrophilic, loves water. Hydro is water, philic means love, right? Because remember, most things are water. In our human body, 65% of our body is water, right? Blood is water, all body fluids essentially based on water. Inside the cell, in the cytoplasm, it's mostly water. Outside of the cell, it's mostly water. So it makes sense that hydrophilic interactions would be the most important. But however, we need to have some of the hydrophobic or water-hating or water-repelling chemistry somewhere as well. And this happens to be these lipid tails facing each other inside the phospholipid bilayer. Now you might ask yourself, why do we need such complexity here? Why couldn't we have just simple one layer? Why create all these different interactions that add that extra level of complexity? And even though it's true that nature generally wants to create things that are relatively simple, because it usually requires less energy, in this case, this level of complexity was absolutely necessary because of how important to keep in mind one main concept, and it is that our cells need to shield themselves from the outside world and have to allow molecules to be only selectively permeable through the membrane, meaning only certain molecules must enter the cell and exit the cell only by permission. We cannot have a free flow of molecules. We can't just let toxins and poisons, all kinds of garbage to enter the cell, and we cannot good molecules that we need like glucose and sugars and others to just escape out of the cell whenever they feel like. Because of that, to create that very strict boundary where only select molecules are allowed to enter or exit the cell, we have created this chemistry. So essentially think about it like this. So now this box here represents that only area where in the cell a molecule will ask for permission to enter or exit the cell. Okay, so if this molecule did not exist and everything was just a double layer, then molecules larger than water would not be able to enter. So why would this be? So think about any molecule. Let's say it wants to enter. So let's say it mixes in together. Let's say it's a hydrophilic thing. So okay, it's happy with the hydrophilic. Can it pass through the fat layer? Again, remember, right? So hydrophilic molecules cannot interact with the hydrophobic molecules because, again, of that rule, like we said, oil and water doesn't mix. So basically, that molecule will be stuck here. If it's a fat molecule trying to pass, again, it can't go past the hydrophilic layer. Right? So any way you cut it, there would be no way for the molecules to pass through. Unless, again, they're extremely small and potentially, if they're like one atom size or something like that, they can squeeze through. But most molecules that we deal with are much bigger, much more complex, and so they would be stuck. So how do we solve this problem? We create these integral proteins, also called channel proteins, that essentially a molecule comes in, asks permission here. These are very large proteins. And the molecule, the integral protein, if it recognizes this is the correct molecule that the cell must accept, it will open its doors for a brief moment of time, for something like 1/1000th of a second, let the molecule pass, and quickly shut the doors behind it so that no other molecules can sneak in behind it. Same thing happens when molecules want to exit out. Now, when you're seeing these words, integral proteins and channel proteins, the only difference between them is that when you're thinking of something like electrolytes like sodium ions, potassium ions that we mentioned already before, usually you think of channel proteins that will interact with them. And general categorization for these molecules is that they're just integral proteins because they span that double membrane and allow for molecules to enter only by permission. Now, the next part of this chapter, let's talk about a little bit how the rest of the cell transport actually takes place. So, when you think of cellular transport, you're essentially thinking of how do molecules pass back and forth through the membrane, how do they enter the cell, exit the cell, how do we categorize this? What do we call these processes? So there's essentially two main type of transports on the cellular level, and this is the passive transport and active transport. When you hear the word passive, what that means essentially is that molecules that are passing through the membrane do not need energy to pass, right? They do this slowly and without any use of energy, right? So main two examples of this is diffusion and osmosis. Diffusion essentially refers to solute or salt molecules passing across a membrane down the concentration gradient. It depends on the size of the molecule that's diffusing and the temperature that the whole state is undergoing in. So, for instance, if the molecule is small, the smaller it is, the easier it is to pass through the cell membrane, and so diffusion will be occurring a little bit faster, the smaller the size of the molecule. Again, the rate of diffusion depends on multiple factors. One of them is the size of the molecule. The smaller the molecule, the easier it is for it to pass through, and so the rate of diffusion increases. And also temperature. If you're increasing the temperature, you're making the molecules vibrate faster, move at a faster rate, and increasing the chances that it will bump against the openings in the cell to pass through. Okay, so the higher the temperature, the faster the rate of diffusion. The smaller the molecule, the faster the rate of diffusion. Again, keep in mind that diffusion refers to molecules such as salt to pass through, not water-type molecules. If you're thinking of solvents or water, now there are many different types of solvents. Something like alcohol could be a solvent, for instance. In biology, water is the primary solvent we deal with, something you can remember that dissolves things. So when water moves across the membrane, we talk about osmosis. Again, this is essentially just like diffusion, except here we're saying water is moving across a membrane. And again, we call this process osmosis. It is also a passive form of transport. Now, in other cases, we need the molecules to move very quickly. We need to add energy to kind of speed up that process of transport across the membrane. In this case, we'll call this active transport. So energy is always required in this case. So just those channel proteins we talked about. So if you're trying to pump sodium or potassium across the membrane very quickly, which is often required in many cells, like cardiac cells or nerve cells and others that we'll talk about later in the semester, you need to add energy to the pump rate to make sure those electrodes are pumped across very fast, and so this would be an active form of transport. Other active forms of transport is something like endocytosis or exocytosis, and these are quite a bit more complex. Essentially, you're thinking about if it's endocytosis, endo meaning inside, this is when a cell is trying to swallow something very large. We'll see this with macrophages, which are very common cells in the immune system as part of the defense mechanisms of the body to protect us against bacteria and viruses and other types of microbes. And if these cells are trying to swallow, let's say, a bacteria, the process will be essentially endocytosis. It will surround that object, that microbe, whatever that might be, kind of engulf it into the cytoplasm of the cell. Okay, so that would be endocytosis. And if the cell is doing the opposite, if it's trying to kind of spit out some kind of a object from inside the cell to the outside, it will do exocytosis, right? Kind of the object will exit out from the cell. Again, they're very similar because so much of the cell membrane is involved and the overall cell needs to be doing this. Obviously, this requires energy, and so endocytosis and exocytosis are more complex processes. They're definitely an example of an active transport. Now, when you're thinking about our cells, most importantly, you'll be concerned about what kind of structures, what kind of components of the cell allow the cell to survive. We already mentioned the nucleus, right? That's the main organelle. Every single cell in the body, with only one or two exceptions, will have a nucleus as long as the cell is alive. Under the microscope, if you see a cell that does not have a nucleus, this is usually one of the first signs that the cell either has died or has suffered tremendous damage. Again, most cells of the body, as long as they're alive, will have at least one nucleus. It's possible for a cell to be also multinucleated, where they have more than one nucleus, and we'll get into these examples later on. Now, so the nucleus has its own nuclear membrane. Inside of the nucleus is the DNA, the genetic material. Remember, DNA is the deoxyribonucleic acid. That's the genetic code that stores all the information to make us who we are. And this is true for all living things, from the smallest, from the simplest to the most complex. All need to have at least some DNA in order to survive, in order to live, in order to reproduce. Inside the nucleus, sometimes under microscope, you also see this another small circle, and this will be the nucleolus. The nucleolus contains the RNA, which is the sort of helper molecule that assists in function of DNA and reading of the genetic code. Now, okay, so we're good. We have our DNA. We know where the genetic information is. Now we step out into the cytoplasm. That's the space between the cell membrane and the nucleus where all the rest of the organelles are. Now, unfortunately, under regular light microscopy, we cannot really see many of these components. You would see them if you're using electron microscopy or magnifying considerably larger, but we still know all of these components exist. When you're trying to figure out what they look like, make sure you're following the illustrations in our textbook to see the pictures of these organelles, to see their general structure. It's important to not only know the names of the organelles, but also their locations and their general overall appearance, which sometimes is helpful to understanding and remembering what they're actually doing in the cell. Now, in order for the cell to survive, to do pretty much anything, to undergo any process, any function, the cell needs energy. The most important provider of energy for the cell is the mitochondria. Now, mitochondria often has this kind of appearance. I'm going to draw for you. Often has this oval with these kind of squiggly lines inside. It often will be red, actually, in color, although not necessarily. And mitochondria also has these multiple membranes inside of the mitochondrial membranes. And the machinery inside the mitochondria allows a cell to generate ATP, or essentially energy. ATP is the molecule that acts as energy currency in the cell. Okay, so the cell will have more and more mitochondria if this cell is constantly active, is doing a lot of things such as muscle cells, heart cells, other cells like that. And overall, these mitochondria will be found throughout the cytoplasm. Another very important type of organelle in the cell is the endoplasmic reticulum, which you can abbreviate as either SER, smooth endoplasmic reticulum, or RER, rough endoplasmic reticulum. Endoplasmic reticulum appears as sort of like a stack of pancakes, which is also membrane-bound organelles, that basically acts like a factory that uses sort of a conveyor belt kind of a style mechanism to process different molecules. So when you're thinking of RER, this is where you're going to see proteins are, after they've been created, they are modified, different molecular groups are added to it, maybe some of them are processed differently, folded, or changed in other chemical or physical ways. So again, rough endoplasmic reticulum, it's rough because ribosomes, which we're going to talk about soon, are actually sitting on top of these membranes, and rough ER is located close to the nucleus. So after the proteins have been synthesized or made, they will enter the RER to be processed further and modified, because as we said before in the previous lecture, proteins are very complex molecules. In order to add all those amino acids and set up the protein for the correct position to function in the cell, quite a lot of different modifications need to be done, and RER is doing that. For the SER, you're thinking of fats, lipids primarily. After they have been made, they need to also be processed and modified to some extent, so that happens in the smooth endoplasmic reticulum. Okay, so what is the difference between them? One is the location. RER is close to the nucleus, SER is further away from the nucleus. RER is rough because it has these dots, the ribosomes on top of them, which look like either white or black dots in some images or illustrations. And SER does not have the ribosomes, so it's a smooth surface without the dots. Okay, so again, RER, you're thinking of protein synthesis, protein modifications. SER, you're thinking lipid synthesis, lipid modifications. You're going to hear the word synthesis a lot here. This is essentially referring to when a cell is synthesizing something, it means it's making something, some kind of molecule. So protein synthesis means making protein. Lipid synthesis means making fats or lipids. So This molecule that we mentioned already, ribosome, this is actually the smallest of the organelles that we have. Look essentially just as dots, but in reality, if you can really magnify it, you see them. This is an RNA molecule that has been trapped by a protein, and it's basically reading the RNA, trying to transcribe the genetic code into a protein molecule. Okay, and that's the one that's actually responsible for connecting those individual subunits of amino acids to create a protein molecule that we need. Okay, so ribosomes are extremely important in the cell. They're active all the time. They could be found throughout the cytoplasm, kind of as free-floating, and many of them are attached to the endoplasmic reticulum, like we just already mentioned. Again, ribosomes start the process of protein synthesis, and then if they're connected to RER, the process continues as modification of those molecules. Now, another question we can ask ourselves, well, how does the cell have that specific shape that it has? How the organelles know where to be, how are they supported, right? So just like a building needs its structure, it's just like the beams and the overall architectural components, and like our body needs the bones and the skeleton, well, our cells need a cytoskeleton to provide the framework and overall shape to the cell. So the cytoskeleton components are actually a different set of proteins, such as microtubules, intermediate filaments, and others that you will see discussed in the chapter. And they are connected throughout the cytoplasm to different components, to different organelles, and allow molecules to freely move throughout the cell to different areas and to provide the overall shape and structure to the cell. So they're extremely important, and we'll see that some of these cytoskeletal components will be used in mechanisms of muscle contraction and other kind of areas where something needs to move throughout the cell. Again, cytoskeleton, very important, a set of proteins to provide the structure, stability, and allow for movements inside the cell. Now, as you're thinking about this, you also might realize that, well, as the cell is doing its job, it might generate something dangerous. Or if it encountered something dangerous outside and somehow got inside the body, how are we going to get rid of these things? So we need an area where we'll have digestive enzymes to break apart those toxic or poisonous materials. This is where lysosomes come in. Lysosomes, again, are membrane-bound organelles found inside the cell in different quantities depending on what kind of cell in the body it is, what organ it's found in. But overall contains a set of different digestive enzymes inside the lysosome in order to break apart different molecules. So if a cell, let's say if a white blood cell in the blood that protects us against microbes, swallow the microbe, that microbe will be essentially incorporated into the lysosome, where the digestive enzymes will try to destroy that microbe to make it harmless for the body, so it's not killing the cell. Okay. In the same kind of method, but slightly more specific, we have peroxisomes, which also have similar to digestive enzymes, except here these enzymes are using peroxide type chemistry to influence different reactions. This does not necessarily have to be destruction of microbes. This could be simply peroxide type reactions done on lipids, for instance, to create certain molecules or modify them. So not a very common organelle and not as important as lysosome, but still exists in certain cells, especially thinking about, again, something like liver cell, hepatocytes, which are known to be detoxifying things and have very significant enzymatic activity in the body. Now, suppose your cell wants to package something to go out of the cell, right? Let's say it's a cell in a gland secreting hormones. How is it going to do that? Also, we have something called the Golgi complex or Golgi apparatus. And this is very similar to smooth endoplasmic reticulum, essentially identical in appearance. The only difference is that instead of what the SCR is doing, what Golgi is doing is taking a molecule that's been created, packaging it to essentially be shipped out of the cell, okay, like a shipping center. So best examples, we're thinking about hormones that need to be modified, need to be packaged properly to be released out of the body. Remember, hormones are essentially small molecules, small messengers that a gland will secrete into the bloodstream to go to distant locations to influence different functions in the body. So a very important component for these cells would be Golgi. Again, in appearance is just like SCR, also located close to the cell membrane, kind of far away from the nucleus, and its job is to prepare and ship out kind of different molecules out of the cell. Okay, and the last component here is centrioles, and this is essentially only visible when we're thinking about cell division, when the cell needs to split in two cells, let's say during mitosis, cell division, which we'll talk about later in the semester. The component of microtubules from the cytoskeleton is sort of borrowed here, and that makes up the centrioles. It creates kind of this mitotic spindle that attaches to the DNA to move it to different spots when the cell is undergoing cell division. Again, you'll see more of this when we talk about this in the later in the semester, okay, in a different chapter. Okay, again, as you're studying this, make sure to read this chapter and pay attention to all the illustrations for these different concepts, especially cell transport, what the cell looks like, what the different components look like. They often are shown in different colors, comparison of animal cells and other types of cells, paying attention to what these different components do in the body.